Good morning, student. Today we discuss about the next cell organelle, ribosomes. Ribosomes is a granular structure. Ribosomes are granular structures first observed under electron microscope. First observed under electron microscope as a dense particle by George Palade in 1953. Ribosomes are composed of RNA and proteins. Ribosomes are composed of RNA and proteins. Ribosomes are not surrounded by any membrane. Means the ribosome cell organelle is a without membrane bound cell organelle. Eukaryotic cells. In eukaryotic cells, ribosomes are ATS type. In eukaryotic cells, ribosomes are ATS type, while in prokaryotic cells, ribosomes are 70S type. In eukaryotic cell, ribosomes are 80S type, while in prokaryotic cells, ribosomes are 70S type. Each ribosome is formed of two subunits, that is ATS type of ribosomes are formed of 60S and 40S subunits. Then 70S ribosomes are formed of 50S and 30S subunits. Here S stands for cell unit and S stands a sedimentation coefficient. In eukaryotic cells, ribosomes are present in mitochondria, plastids, in plant cells, in cytosol. Ribosomes are either found attached to outer surface of rough endoplasmic reticulum, outer surface of rough endoplasmic reticulum and nuclear membrane. Or free suspended in or free suspended in in cytoplasm. Free suspended in cytoplasm. Bound ribosomes. Bound ribosomes generally produce proteins. Bound ribosomes generally produce proteins that are transported outside the cell. That are transported outside the cell after processing in endoplasmic reticulum. After processing endoplasmic reticulum and Golgi body. Example of bound ribosomes. Bound ribosomes of acinar cells of pancreas. Acinar cells of pancreas. Acinar cells of pancreas produce pancreatic digestive enzyme. Produce pancreatic digestive enzymes. Free ribosomes. Generally produce enzymatic protein. Free ribosomes produce enzymatic proteins that are used in cytoplasm. Like enzymes required for breakdown of sugar. Both types of ribosomes, that is the bound and free types of ribosomes, can interchange position and function. Number of ribosomes is high. In cells actively engaged in protein synthesis. This is about the ribosomes. Then second cell organelle is the nucleus. Next cell organelle is nucleus. Structure of nucleus of eukaryotic cell. Structure of nucleus of a eukaryotic cell become distinct eukaryotic cell become distinct in a non-dividing cell. Distinct in a non-dividing cell. Distinct in a non-dividing cell or during interphase or during interphase. Interphase nucleus is made up of interphase nucleus is made up of nuclear envelope or nuclear membrane. Second, nucleoplasm or karyolim. Third, nucleolus. And fourth is the chromatin network. Chromatin network. First, nuclear envelope. First part, nuclear envelope. Nuclear envelope is a double-walled delimiting membrane. Double-walled delimiting membrane. Nuclear envelope is double-walled delimiting membrane of nucleus. Two membranes are separated from each other. Two membranes are separated from each other by perinuclear space. Two membranes are separated from each other by a perinuclear space. This is perinuclear space, outer membrane, inner membrane. outer membrane and inner membrane. These two membranes are separated from each other by perinuclear space. Outer membrane is connected with endoplasmic reticulum at places connected to endoplasmic reticulum at places and harbors ribosomes on it Inner membrane is aligned by nuclear lamina. Nuclear lamina is a network of protein fibers that help in maintaining the shape of nucleus. The two membranes, that is the outer membrane and inner membrane. Two membranes, that is outer membrane and inner membrane with perinuclear space help in separating nucleoplasm from cytoplasm. Nucleoplasm from cytoplasm. Nuclear membrane is not continuous. There are a small opening called nucleopores. Nucleopores on the nuclear membrane. The nucleopores guard by pore complex which regulates flow of substances from nucleus to cytoplasm in reverse direction. The nucleopores are guarded by pore complex which regulate flow of substances from nucleus to cytoplasm and cytoplasm to nucleus, that is in reverse direction. Then second part, nucleoplasm or karyolim contains various substances like nucleic acids, protein molecules, minerals, nucleoplasm or karyolimb contains various substances like nucleic acids, protein molecules, minerals and salts. It contains chromatin network and nucleolus. It contains chromatin network and nucleolus. Then contains nucleolus and chromatin network in karyolimb. Then third part is the nucleolus. Nucleolus is made up of rRNA and a ribosomal protein. rRNA and ribosomal proteins. rRNA and ribosomal proteins, and it is a known site for ribosomal biogenesis. It is a known site of ribosomal biogenesis. The rRNA and ribosomal proteins are transported. rRNA and ribosomal proteins are transported to cytoplasm and are assembled together to form ribosomes. Depending on synthetic activity of a cell, there are one or more nucleoli. There are one or more nucleoli present in the nucleoplasm. Present in the nucleoplasm. Example, cells of oocyte. Example, cells of oocyte contains large nucleolus. Example, cells of oocyte contains large nucleolus. Sperm cells contain small inconspicuous one chromatin network, present near chromatin network. Then next part, chromatin network or chromatin material. Chromatin network or chromatin material. Nucleus contains genetic information in the form of chromosomes. Genetic information in the forms of chromosomes. In the forms of chromosomes. The chromosomes appear as a thread-like. Chromosomes appear as a thread-like network. This network is called as chromatin material. This network is called as chromatin material. The chromatin material contains DNA, histone and non-histone proteins and non-histone proteins and RNA. DNA, histone and non-histone proteins and RNA. In some region of chromatin, in some region of chromatin, in some region of chromatin, DNA is more in some region of chromatin, DNA is more and genetically active This region is known as euchromatin. In some region that contains more proteins, in some regions that contains more protein, and less DNA. More protein and less DNA. genetically inert are called heterochromatic. Are called heterochromatic. Metaphase stage, they become distinct at metaphase. They become distinct and can be clearly visible or identified. Every species of living organism has a specific number of chromosomes. In human cell has 46 chromosomes. Then functions. Nucleus contains entire genetic information. Hence, play an important role in heredity and variation. It is the site for synthesis of DNA, RNA and ribosomes. Play an important role in protein synthesis. Then important in phylogenetic studies. And nucleus is the master cell organelles. Thank you.